morning. My name is Abe Growcott. I work for Biosecurity New Zealand. My job title is Principal Advisor, Aquatic Surveillance. I've been working at Biosecurity New Zealand for well north of 10 years now. I've stopped counting, and it's a real privilege to be here today to deliver this webinar on marine pests and the marine biosecurity system. So let's get into it. So just a brief background on our session today. I'll talk about the marine biosecurity system in general to kind of situate us, then talk about marine pests that are not present in New Zealand, but are high risk. I'll talk about marine pests that are here in New Zealand, but only in localized locations. Talk about how to report any unusual findings, where to find info, and additional resources that you may be interested in getting your hands on. So this is my one heavy slide for the day, team. I apologize, but we'll get it out of the way. This is a schematic or an analogy of the marine biosecurity system. And each of these nets is an intervention in the biosecurity system. And these nets, they catch biosecurity risks. So if we start from the left here, you can see that there's biosecurity risks entering into these nets and they're getting caught. And so this first net is what we call the offshore intervention. And so that's if any person or commodity or conveyance, whether that be a vessel or an airplane, they all have to meet rules to enter New Zealand, and they're called import health standards. So our offshore intervention is getting people to comply with those rules. And if they do that, the biosecurity risks will be mitigated. For example, that might be a vessel entering New Zealand with a clean hull and having low levels of biofouling attached to that hull. That might be a container which has some commodity that we're interested in being fumigated before it enters the country. So this is a very efficient way to mitigate biosecurity risks as it's preventative, and it's very cost effective, and it means we don't have to deal with those biosecurity risks when they get to New Zealand. So if this schematic was a little bit more accurate, the nets on the left-hand side here would be much larger because they catch more biosecurity risks. So that's our offshore intervention. And then behind that, because no system is 100% effective, is that we have the border intervention. And so that is all about our quarantine officers using sniffer dogs. This is what everyone thinks the biosecurity system is Border Patrol on TV with the sniffer dogs looking for people, arriving in the country with apples or bananas, etc. It's all about risk profiling, targeting effort to where the risk is, and it's all about making sure that importers comply with those import health standards. So that's the border intervention. Behind that, if a biosecurity risk does slip through and doesn't get caught by these first two nets, we have the surveillance and incursion investigation net. And this is where the team that I work for, live in the biosecurity system. So this is all about designing surveillance approaches for early detection of biosecurity risks out in the environment, trying to find things very early, identifying them and acting quickly to either eradicate or reduce population density to protect our marine values. If we do find something and we can't eradicate it initially, we'll then surge into the following nets, and that is around responding to a marine pest. And this is where we can really surge with people within Biosecurity New Zealand and wider organisations to assist in and trying to eradicate a marine pest or disease, or slow its spread, mitigate those impacts. And finally, we have the last net, and that is pest management, so long-term management of an organism that we can't eradicate at this stage, whether the technology doesn't allow us to do that, etc. And it's all about trying to protect local high-value locations and limit those impacts to humans, the environment, and the economy. So today's talk is around this outcome here, where everyone is a champion for the biosecurity system and owns their responsibilities and knows what to do, has the knowledge and the motivation to do the right thing, and so that we can all work together. So I hope this kind of schematic at the start of this presentation just helps situate us in the biosecurity system, and to show that there's a whole lot that happens before things come into the country and a whole lot of things that happen after. And it's not just Border Patrol on TV with the sniffer dogs. So this is the team that I'm in. We are aquatic health team, so we're managed by Mike Taylor. We have 3.5 incursion investigators. Many of you have probably engaged with the incursion investigators before. And we have 2.5 surveillance advisors. So Julia is 0.5 here of a incursion investigator and 0.5 of a surveillance advisor. So that's our team. We're mainly situated in Wellington, but Josh has recently moved to Auckland, and that's really helped because a lot of the action happens, you know, in the top part of the North Island. So the main marine pathways for pests to enter New Zealand are listed here. So we have shipping, whether that is a commercial vessel or a recreational vessel coming into New Zealand. Marine pests can enter via ballast water or as attached to as biofouling on a vessel hull. We also have what we call topside risks with vessels, and that's a good example for that is exotic caulerpa, where we think that it may have come in attached to a contaminated anchor or anchor chain. So it's not just ballast water and biofouling, there are definitely topside pathways too on vessels. We also have aquaculture. Species can't be imported into New Zealand to culture, but there are rules around that, and equipment. And then we also have the ornamental fish trade. So these are the main pathways for organisms to enter the country. An analysis was done a number of years ago looking at the non-Indigenous marine species present in New Zealand and their different life stages, and it was surmised that biofouling is definitely the predominant pathway for those pests that have established historically in New Zealand, and it was estimated that up to 90% of those species that are now present in New Zealand have a life stage that could allow them to enter into New Zealand attached to a vessel hull. So moving on to the marine pests and the how to ID them phase of this presentation, I thought it would be quite useful to talk us through the 5 species that the National Marine High Risk Site Surveillance Program was designed to detect. So this is a surveillance program that occurs in the 12 busiest ports around New Zealand since 2002. And it is operationally completed by NIWA on behalf of Biosecurity New Zealand. And these are the 5 species. So we've got, and these, just to be very clear, are not known to be established in New Zealand. So we have in the chat there a link to the Marine Pest ID guide. So that's where these pictures are from. So if you have a second screen and would like to look at the Marine Pest ID guide and zoom into the pictures with a bit more resolution, feel free to click on that link if you can see it. And you can see the page number there. I've got them listed for each of the pests we're going to look at today. So we'll start with aquarium caulerpa. So this is Caulerpa taxifolia. This is not present in New Zealand. It has fronds, which are about 1 centimeter wide and 15 centimeters high. And they are attached at 90 degrees to these horizontal runners that are up to 9 meters long. Just to note that this looks very similar to exotic caulerpa, which is present in New Zealand, and we'll discuss exotic caulerpa a bit later. Aquarium caulerpa is really bright green. It's unnaturally green. It looks unnatural. And then there's these on the fronds, each of the fronds that grow at 90 degrees from the horizontal runners. There's this midriff, and you can see the number 2 there on the picture. So that's an identifying feature. There's this midriff, and then from that there's these little leaves called pinnules that are attached to the midriff that grow off horizontally up that midriff, and it gives it this feathery appearance. These pinnules or leaves are flattened, and aquarium caulerpa, as you can see with the little sign, has not been found to be established in the marine environment. And the most likely way that this could enter is via someone releasing their marine aquarium contents into the environment. Aquarium caulerpa could be a hitchhiker that could enter via the aquarium trade. It's not permitted to be imported into New Zealand, but there have been historical finds of the species in the past that have been destroyed. This species is present in Australia, so New South Wales, and most famously, it's established in the Mediterranean, where it covers a large amount of the Mediterranean now. Moving on to the Chinese midden crab. As the name suggests, it has these hairy middens on its front claw with white pincer tips. So that's probably one of the dead giveaways for this species. It also has a distinctive notch between its eyes. That's number 2 on the picture there. And it also has 4 spines on each side of its shell or carapace. And that's number 3 there. You'll see its hind legs there. It doesn't have paddles on its hind legs. Our native paddle crab, as its name suggests, has paddles. So that's another identifying feature. So yeah, you've got the hairy middens, the white claws, the notch between the eyes, the 4 spines on the side of its shell. The carapace is about 10 centimeters wide. So it's not a terribly large crab, but it can reach really high densities overseas. So this crab is present in Europe and in the UK. And it lives in freshwater environments and marine environments. So they'll live in rivers and stuff and burrow and get to really high densities and create many burrows and riverbanks. And that can cause a lot of erosion and sedimentation because these burrows can collapse, can cause issues for infrastructure in the rivers. And so yeah, these crabs live in the rivers, and then when they come down to breed, they all march down together and go into estuarine environments to breed. So this can be a lot of biomass moving between those 2 systems. It's most likely to enter New Zealand via vessel biofouling or ballast water. Here we have the Northern Pacific sea star, Asterias amurensis. This species is present in Australia, in Port Phillip Bay in Melbourne, and also in the Duwart estuary by Hobart in Tasmania. It's been present in Australia for well over 20 years, and it's reached very high densities there. It's got 5 arms, and they are pointed and often upturned. So that's a key identifying feature, the 5 arms with the upturned ends. They can vary in their coloration, so they can be yellow morphs and also more purpley yellow. The arms on top of the sea star are covered with these chisel-like spines, and they're randomly assorted on top of the sea star. So a lot of sea stars have these spines in rows, whereas this species has them randomly placed. This is quite a big sea star. It can reach kind of dinner plate size when the really, really big ones. So yeah, they can be quite large. And in estuaries or embayments that are large and are not subject to a lot of wave action, this species can reach really high densities. So in Port Phillip Bay, it's reached very, very high densities. You can see the picture, those 2 pictures with the number 6 on them. That's pictures from Australia, and these guys kind of move across the seafloor, eating whatever they can get their hands on, or their arms, whatever they get their arms on, they can consume. So shellfish beds, and other organisms. So we definitely don't want this species here. And then we have the European shore crab, Carcinus manus. This species is kind of a medium-sized crab, so the shell is about less than, quite small actually, 8 centimeters in length. Again, the key to ID this species is the number of spines on each side of the shell. It's got 3 rounded teeth or lobes between its eyes. It looks similar to New Zealand's native paddle crab, Ovalapes catharis, except if you look at the number 4 there, its hind legs also doesn't have doesn't have paddles. So again, that's a key distinguishing feature. Juveniles are generally lighter in color than adults. And again, this species can reach very high densities. You'll find with a lot of these pests in the right conditions, they can reach very high densities. This species is present in Australia and in multiple locations. It can reach very high densities. It can consume a lot of biomass in the marine environment. So you can imagine in a reef system in New Zealand, if this got up to high biomass, it would be consuming a lot of food and impacting the ecosystem. And this is our final species for those that are key to look out for, but are not known to be present in New Zealand. This is the Asian clam, Potamocorbula amurensis. So this is not the golden freshwater clam. This is a different clam. This is a very small bivalve. It's only about 3 centimeters in length, but it can reach incredibly high densities. The key identifying feature here is that one of the 2 shells is bigger than the other. So it creates this very, very distinctive overbite. So that's the biggest giveaway for this species, is this overbite. So it's a very small size. It has this overbite. The shell can be very thin and smooth and almost translucent if you find the really, really small one. In the older individuals, the shell can be dirty white, tan, or yellow with this kind of brown staining. This species, to my knowledge, has only been detected outside its native range in San Francisco Bay. There it's reached, like I said before, extremely high densities. And as it's a filter feeder, you can imagine the plankton and phytoplankton, it would be filtering a lot of that out of the water and impacting the ecosystem. It's been thought that the introduction of this species in San Francisco Bay reduced a number of important recreational and commercial fish species. So it's definitely a species we don't want here. It inhabits muddy and sandy habitats, so that's where it would most likely be detected. So yeah, they're the species that the Marine High-Risk Site Surveillance Program is designed to detect. And so now we're changing tack now and looking at species that are present in New Zealand. I just like to acknowledge that there are other species that, for example, regional councils, etc., have on their pest management plans. But I didn't want this list to get any bigger, so I've just restricted it to these species. But I just like to acknowledge that there are other species of interest. So here we have exotic caulerpa. So this consists of two species. So we've got Caulerpa brachopus and Caulerpa parvifolia. For all intents and purposes, they look exactly the same. So they look exactly the same. The species determination isn't as important. So that's why it's called exotic caulerpa. It looks pretty much exactly the same as the aquarium caulerpa, Caulerpa taxifolia, that we were looking at before, except that the frond that grows from the horizontal runner, it's solid in shape. It's oar-shaped and it's solid. It doesn't have the midriff and it doesn't have those pinnules or leaves that run up. It's just completely solid, and that kind of highlighter green. So yeah, no other. We have native um, native Caulerpa species, and none of them have this runner and frond morphology. So if you see this this morphology, please let us know. Uh, no native Caulerpa looks like that. So that's the key. You've got the horizontal runner with the fronds growing at 90 degrees. We can see that exotic Caulerpa has been detected at a number of locations in the Upper North Island on the east coast there. And yeah, the fronds can grow in quite similar size to aquarium Caulerpa, about 10 centimeters in length. Here we have the Asian paddle crab, Charybdis japonica. This is quite a large crab. It has 6 spikes on each side of the carapace. So on the shell there, so that's number 1. And then another key distinguishing feature is that it actually has spines on the upper surface of each claw, so it has 5 of them. So you've got the five spines on the upper surface of the claw, 6 spikes on each side of the carapace or shell below the eye. This species does have paddles on the back legs, so it's those spines that are key for identifying this species. There's also different color morphs, as you can see there. There's kind of an off-white and pale green, olive green or brown crab, and then another one with purplish markings. The carapace can be up to 12 centimeters wide, so it's quite a large crab. Behaviorally, this crab is quite aggressive, so that can be another way to not ID the species, but to, see that this is something unusual. Our native paddle crab normally just wants to get away from big humans, whereas this one can stand its ground, rear up, and show us its big claws, and apparently it gives a nasty bite. This species has been present in New Zealand for around 20 years from memory now. Yeah, and it's slowly spreading south. So on the east coast, it's been found as far south as the Bay of Plenty now, and on the west coast, it's been found as far south as Raglan. Here we have the Mediterranean fanworm, Sabella spallianzii. And the key feature here is the single spiral of the fan of the feeding tentacles. So when it feeds, it has these beautiful looking spiral fans that protrude from the top of the tube, and they have a spiral look. Our native species don't have that. They also have a large livery tube. So the tubes can be 10 to 50 centimeters long, but they can be in some cases up to a meter long. So this is by far the largest fan worm in New Zealand. Our native species are much smaller. The livery tube is brown grey. It can be finely banded with concentric rings and can be muddy looking and livery. Um, our native species, the tube will be connected to substrate, not just at the base, but along the side of the tube. So it's almost like the fan worm is stuck to a reef or a rock or whatever it's attached to, whereas the Mediterranean fan worm is only attached to substrate at its base. So that's another way to tell. That it is Sabella. This species was first detected in 2008 down in Littleton, and a response was initiated, and that population was either eradicated or reduced to a very, very low density for a number of years. Subsequently, it was detected in the Waitemata not too long after, and it was found that it was too widespread to effectively manage. So it's found in a number of locations now, but is being actively managed when it is found in new locations. Here we have the club tunicate, Styla clava. So this is a brown-looking ascidian with lumpy, warty, livery skin, kind of woody in look. It has a tapering stalk, which is often longer than the body of the organism. Has these two kind of closely spaced siphons at the top of the body. So that's picture the numbers 3 there. And yeah, usually surrounded by warty projections. It's got that club shaped, hence the name, and can be up to 20 centimeters in length. And this species is found at a number of locations around New Zealand now, can reach really high densities, can filter a lot of water, a lot of plankton and phytoplankton out of the water. So it can be impactful to the ecosystem and also a nuisance species to aquaculture. Here we have Undaria pinnatifida. So this species was first detected in New Zealand in 1987 in Wellington Harbour, and again is found in a number of locations now around New Zealand. This is a large macroalgae, typically less than 3 meters in length. It's brown to yellow, green in color. It has this this organ called a sporophyll, frilly sporophyll, near the base of the plant, and that's actually its reproductive structure. So that looks like lasagna. So that's a very key identifying feature, is this sporophyll, this lasagna-looking organ at the base of the plant. It also has a midrib, which is that midrib, which is number 1 there in the picture. And then these smooth, thin leaves that protrude from that midrib, and it stops short of the base. So there's always a gap between the bottom of the leaves and the bottom of the plant. In summer, Undaria, due to high water temperatures, can kind of die back, where the whole top of the plant erodes or dies away, and all that you see is the sporophyll, and then that plant can regrow when when the water temperatures increase again. So this species loves disturbed habitats, and yeah, is a fowler, so can be spread by vessels that aren't adequately managed, as you can see by that picture there in the middle. And finally, we're almost there. This is our last species, team, so you've done really well. We have the Australian droplet tunicate. So this is Eutostoma elongatum. This is it's as you can see, looks like white sausages. It has a white or cream coloured tubes. They're generally about 5 to 20 millimeters in width, and they can be quite long, typically around about 5 to 30 centimeters in length, but in the right conditions, they can be up to 1.5 meters long, these kind of white droopy sausages. The tubes are actually colonies of individuals, which is, I reckon, pretty cool. And when they're carrying larvae, they can have these orange flecks in the tubes. So pretty distinguishable. They tend to grow really prolifically in summer periods when water temperatures are warm, and then they tend to die away over winter, or if there's a big flush of fresh water into the coastal environment. As you can see, found in the upper half of the North Island, and yeah, has spread south a bit as those marine heat waves have occurred. So that is all the marine pest part of the talk done. So well done, team, for making it this far. And I'd just like to talk a little bit about the notification process and all of that. So the key here is if you remember nothing about what I've talked just before, this would be the key slide, is to just report the unusual. So we have the 0800 Pest and Disease hotline. You can see the number there. And we also have an online notification system. So if you're out on your bushwalk or you're walking down, on the beach with your dog, the key is to report the unusual. You don't have to know what the species is. You just have to know that it's different. So you guys know your environment better, really, really well. So report the unusual. You can call this number. A lot of the time, an ID can happen from a really, really good photograph. So if you've got your smartphone and you can take a real good bunch of photos, that can be enough a lot of the time to ID a lot of different marine pests. So that's the key message: report the unusual. This is just a very quick kind of process around what happens when you do engage with Biosecurity New Zealand. So we have our surveillance, whether it's a targeted program run by central or regional government or other groups. And then we have our general surveillance, which is all of us, the public, out and about doing our thing. So we have all these different notifier groups. And then we have our channel, so our 0800 number or our online notification. You call us or you email us, it gets triaged, directed to the appropriate team. For this talk, it's Team Iman Aquatics. And then we have an incursion investigator. So they're on call every day of the year from 8:30 a.m. to 8:30 p.m. And they are required to respond to that notification within 30 minutes. So that's the generalized process. So why are we doing this? It's to avoid that situation in the picture. So that's, as you can see, Chinese midden crabs in the UK. You can see that whole picture is pretty much just them. So yeah, you can see the densities that these organisms can get to outside of their native range. They can get to very high densities and would be consuming, a lot of food to maintain that biomass, which is, yeah, a situation we want to avoid. So I quite like this little this little schematic here where we have a population of non-Indigenous species represented by this red line. And on the bottom axis, the x-axis, we have time since establishment. So closer to the left there is less time since the organism got to New Zealand, for example. And then we have cost to manage. And that red line is the population size. So what we're trying to do is to detect these new species basically as soon as they get here, so that they're at lower population number and at fewer distinct locations. So the earlier we can detect on this line towards the left-hand side, the cheaper it is to manage the cost, whether that's a human cost, monetary cost, an ecosystem cost. It's a lot lower. So we're trying to get the detection down as close to time zero when that organism entered the country as possible. And then we have more options. If we detect higher up this line, the population may be too high. It may be the species may be in a number of locations and it's not feasible or practical, regardless of how much money you throw at it to eradicate that species. So yeah, the earlier we detect, the more options we have. And this is where you can be a big help. I put this slide in here. It's more geared towards disease diagnosis, but we can help if you ring the 0800 number. You don't have to know how to package up organisms, but we can help you. We can help you source materials, give you methods, and a lot of instructions around how to get organisms to where they need to be to get identified or tested. The key is specimen quality, and this is true for disease diagnosis of species and also for identification of pests, especially if they need molecular testing to be paired with a morphological assessment. So the best I've ever heard of ideal specimen quality is that the specimen you're submitting should be good enough to eat, you know. If it's not, then it's really hard, especially for disease diagnoses, to find out what killed that organism, especially if it's contaminated with secondary bacterial infection, for an example. So yeah, getting good, fresh, quality samples is really, really important so that we can figure out what's going on. When submitting samples, depending on what type of analysis is done, it can take a while. For marine pests, molecular testing can take a number of weeks. And it's not just the testing, it's not just the sequencing, it's also the analysis, looking at the phylogenetic trees, trying to figure out what species it is. It all takes a lot of work. But the incursion investigator will keep you in touch and update you with any progress. And then where does all this stuff go? Where does all this data go? Well, Biosecurity New Zealand, every 3 months, publishes the surveillance magazine. So that's four publications a year. And there are all these details of the latest investigations and the outcomes of them. So they can be a really good read to understand the historical investigations and the outcomes of recent ones. For marine pests, we also have the Marine Biosecurity Porthole, which has mapping, different maps for different for marine pests, and you can search a specific marine pest and figure out where it is in the country, when it was found, etc. So please feel free to go on there and have a look around. And then finally, I just wanted to highlight that we have these general surveillance resources available. If you're interested in some of these resources, please flick me an email and we can provide them to you. Probably the most relevant is the Marine Pest ID Guide in the center here, and these little pamphlets here that summarize what you need to do when you report aquatic disease or an aquatic pest. they're a really useful little cheat sheet to have on hand. And if you have any ideas around surveillance resources that you would like, please get in touch. We're always open to new ideas and new ways to engage. So please let me know. Thank you very much. And yeah, any questions? Cheers.
Thank you, Abe. We did have some questions in the Q&A box. So anyone who has questions, feel free to drop them in there or put your hand up. One question from Gabriel says, does the water temperature significantly affect the likelihood of these pests establishing in New Zealand?
Yeah, depending on the pest, of course, but definitely the establishment likelihoods can increase for a number of species with increasing temperature. Yeah.
Okay. Beth asks, well, she's actually put in two questions, so I'll group them together. She says, what should we do if we find Mediterranean fanworm? Do we pull it out and destroy it? And she's asked a similar question for the Australian droplet tunicate. How should she try and get rid of these species?
Yeah, it's hard. With Ascidians, a lot of the time they can survive fragmentation. So you have to be very careful if you were going to remove them. The best thing you'd do would be to give us a call so we can figure out where you are and whether it's established there or not. And then figure out whether it's worth actually removing in the first place. That would be the first step. Mediterranean fanworm, my colleagues at NIWA call it the dog poo method, where you get, because Mediterranean fanworms, they can eject from their tube when they're disturbed as a survival mechanism. So I've seen videos with many Mediterranean fanworms on the hull being brought out of the water, and as it's being as the hull's exposed to air, they'll actually drop out of the tubes back into the water, so they're just left with a tube. So you want to avoid that happening. So they call it the dog poo method, is where you get your bag and you put it up your arm and you actually put it on top of the Mediterranean fanworm and move it down, and then remove the base of the fanworm, and so that it's all wrapped up in a bag. You don't want to just break off the tube, say halfway, because the living part of the fanworm can retract right down to the base and still survive. So yeah, there's a little bit of nuance there around that.
Bernetta has said something about a study that found biofouling as the main pathway. Study, please.
Sorry, I missed that one.
Bernetta said, Abe mentioned a study that found biofouling as the main pathway. Can that study be shared with us, please?
Yeah, sure. Yep.
Yeah. Okay, so Bernetta, maybe just send an email to info@marinepest.nz, and I'll ask Abe to send some information. Back to you. Catherine has a copy of the November 2022 Marine Pest ID Guide and asks if it's worth swapping out for the more recent one.
No, they're just minor updates there, Catherine. I can't remember from the top of my head, but super minor, just around some of the little maps for the species that are in New Zealand, just around the little red dots. So it was, yeah, super minor, not worth swapping out.
Okay, another person says, what is the priority for MPI response to the Mediterranean fanworm in locations it hasn't been found before?
Yeah, so we have new locations, definitely to manage it, depending on the situation. If it's found at low numbers and if it's detected early, then we have more options. So it is being actively managed at a number of locations around New Zealand where it is still able to be managed.
And Ollie asks, is there information regarding common depth bands to find some of these species?
Yeah, good question, Ollie. There's a little bit in the Marine Pest ID guide. I probably should have talked about that a bit more. Yeah, but if you have specific species in mind, I can always you can always send me an email and I can dig up information. No worries at all. Noting that some of the species that say we haven't got in New Zealand yet, some of them do act differently or behave differently when they come to new locations. So you should always treat that overseas information with, you know, new species can act different in new environments. So sometimes they can live deeper or shallower or grow faster or reproduce at a different time of the year, etc., etc. So yeah, there can be some plasticity there.
And another question from Beth says, it's great reporting, testing and IDing, but how can we assist with eradication directly? And I know that that's a really big question and but doesn't have an easy answer.
Yeah, there's a few things to step through there with that. It depends on the species, it depends on the scenario, it depends on the methods that we have, it depends on the health and safety, depends on the budget. Yeah, a lot of depends. So yeah, as you said, no easy answers.
Okay. One more. How do you manage fanworm if it's already present at a location?
Yeah, good question. Well, it is an unwanted organism, so you have to get, if you want to manage it at a location, you have to get what we call a Section 52 permission under the Biosecurity Act. But if it was to be managed and you had that permission, it's basically hand removal. Yeah, that's the only tool in the toolbox really for that species, and that's how you go about it.
And I guess there's also a focus on making sure that we don't spread it, right? So
yeah,
it may be
100%.
location. So therefore, it's really important that if we own a boat or are working in the water, that we don't take it with us to new places.
Yeah.
Bernetta says she's referred to Biosecurity Act amendments and a timeline for changes. Can you advise on timeline for changes following the recent consultation, which included questions on pest and pathway management plans, and that was in relation to the Biosecurity Act amendments that was consulted on earlier this year, I think.
Whoa, that's a really, really good question. I can't give updates on the timelines there because I'm honestly not sure. These things can have a winding path. The Biosecurity Act's been amended for many, many years now. So yeah, I'm not sure when that end date, when it's all being kind of tied up and finished. Yeah, so sorry about that.
Great. So thank you, everyone. I think that probably brings us to a conclusion. Thank you very much, Abe, and also to the Top of the North Marine Biosecurity Partnership and the Top of the South Marine Biosecurity Partnership. You can visit their websites, marinepest.nz and marinepest.co.nz, to find out more about the work in those specific regions. Really appreciate everyone coming. The session has been recorded and it will be posted on both websites so that you can take a look. We will email details to you. Thank
Awesome.
you.
Thank you, team. Have a great day.
You too.